The overall goal of this procedure is to demonstrate a simple and highly reproducible protocol for the generation of recombinant arenaviruses in FDA-approved Vero cells for potential vaccine or vaccine vector seed development. This is accomplished by first carefully preparing the plasmid DNA transfection mixture. The second step is to transfect Vero cells with the plasmid DNA transfection mixture. Next, it is necessary to scale up transfected cells into 100 mm dishes. Ultimately, the final step is to confirm a successful virus rescue by immunofluorescence using wild-type virus, or by fluorescence microscopy and Gaussian luciferase expression using trisegmented virus.
Compared to existing methods, such as rescues and murine cell lines, this protocol generates arenaviruses in Vero cells which are approved by the FDA for the development of vaccines.
In addition to the generation of potential vaccine seeds, the reported gene-expressing trisegmented virus can also be applied for the identification of antivirals against arenaviruses in high-throughput screens without the need of secondary assays for viral detection.
First, prepare 250 microliters of optimum media and, depending on the virus rescue, 10 to 12 micrograms of lipofectamine 2000 per transfection. Incubate for 5 to 10 minutes at room temperature. During this time, prepare the plasmid transfection mixture in a separate microcentrifuge tube